Hi, I'm Patricia Pereira. I'm an assistant professor at the Mallinckrodt Institute of Radiology, Washington University School of Medicine in St. Louis, and my project is on modulating receptor trafficking for improved imaging and therapy of neuroendocrine tumors. What attracted me was first the high incidence rates of neuroendocrine tumors and also high resistance associated with this disease. Having a personal experience of seeing what the damages of cancers in the patients, but also in their families, that brought me a sense of trying to do something not only to understand the disease from a scientific point of view, but also to translate the things that we do into the clinics to help patients in terms of their therapies with the goal of improving their survival and quality of life. My project in general terms has the goal of increasing the amount of drugs that gets to the tumor. Cancer cells, they have these receptors on the surface, and so think about these receptors like gatekeepers. And so if we have more of those on the surface of the cancer cells, they will be able to uptake more of this drug. And so what we're doing in the lab is trying to find ways to keep these receptors, these gatekeepers on the surface for a certain amount of time, so they will be able to take up more of this drug, and then they take it inside of the cell and they will release the drug inside of the cell. They will induce DNA damage and they ultimately will kill the cancer cell. The key question we're trying to answer is, can we use drugs that they are already used in the clinics for other conditions? Can we use these drugs to modulate a receptor in neuroendocrine tumors that's called SCSTR2 receptor? And so we use non-invasive molecular imaging approaches to see in the whole body in real time where are these drugs traveling in the body. Then by modulating this SCSTR2 on the surface, we want to enhance uptake of a drug that it's called Lutathera, which is an FDA-approved drug. And so in ways that we will ultimately enhance the amount of drug that gets to the tumor, and then we'll have improved the therapy. We're trying to answer the question of whether or not we can modulate biology in a temporary and a reversible way, so we improve drug uptake and we improve efficacy. From a translational point of view, is for tumors that do not respond to these therapies, can we reverse this resistance? Can we convert them into responders? And for the ones that they are responding, can we improve the therapeutic response for a longer time and reversing resistance? This funding was really important because I started my lab during the pandemic, and so there were a lot of challenges associated with that, and having this vote of confidence from the foundation and from the donors believing in our research at that stage in my career was really important. So I'm very thankful to the donors and for their vote of confidence in our research, and for being partners in us while we try to improve therapies and improve survival for patients.